This podcast, a simple summary of a scientific peer-reviewed paper, is made with the help of AI by Big Head Pain, Dr. Farayadun Hagdoost. Today's episode covers the paper titled Biochemical Insights into Diverse Psilocybe Mushrooms and Their Metabolites as Sources of Neuroactive Agents, a review by Sudhakaran et al., published in Current Microbiology in July 2025. You've probably heard of psilocybin, the psychedelic compound in so-called magic mushrooms. But did you know that some species of psilocybe also contain psilocin, baeocystin, norbaeocystin, and even beta-carbolines, all of which may contribute differently to their effects on the brain? This paper dives into over 300 species of psilocybe mushrooms, comparing their chemical diversity, how they produce their active compounds, and what makes some species more potent or therapeutically promising than others. Psilocybin itself is not the molecule that acts directly in the brain. It's actually a prodrug that gets converted into psilocin, which then binds to serotonin 2A receptors. This receptor activity is what leads to changes in perception, emotion, and cognition. Interestingly, some species produce different ratios of these compounds. For example, Psilocybe azurescens is one of the most potent, with psilocybin content reaching up to 1.8% of its dry weight. Meanwhile, other species like Psilocybe cubensis, commonly used in clinical research, contain lower but more consistent amounts. These differences are influenced not just by genetics, but also by ecological factors, like the type of wood or dung the mushrooms grow on, or the temperature and humidity of their environment. The paper also highlights newly discovered biosynthetic pathways. Instead of following the originally proposed linear process of psilocybin formation, recent studies suggest a branch pathway that allows for intermediate compounds to be shunted into different end products. That's how you end up with minor tryptamines like aruginacin or norpsilocin, which may have milder or modulating effects. The result? A chemical entourage effect that might explain why the subjective experience of different mushrooms can vary, even at equivalent psilocybin doses. And there's more. Some psilocybe species contain beta-carbolines like harmine and harmane, monoamine oxidase inhibitors that may prolong or potentiate the effects of psilocin by slowing its breakdown. This biochemical synergy hasn't been deeply explored yet, but it adds complexity to how we understand and possibly use these mushrooms in clinical settings. Beyond pharmacology, the paper underscores that the taxonomy of psilocybe has undergone recent revisions. Species were once lumped together or misclassified, and without accurate identification, clinical research can't move forward reliably. For instance, what one researcher called Psilocybe semilanceata might have been something entirely different depending on the region and analysis method. In summary, this review shows that psilocybin is only part of the picture. The full range of neuroactive compounds in psilocybe mushrooms, along with their ecological and genetic context, could hold the key to developing safer, more effective psychedelic treatments. It's not just about one molecule, it's about the whole mushroom.